Hello, and welcome to our second Science Cafe of 2024. I'm Fred Bever. I'm the Chief Communications Officer here. Maybe I'll speak a little slowly because I see more people still are coming in to join this MDI Bio Lab Cafe. Welcome, everyone. I hope the new year is developing well, or as well as possible, given all of the circumstances of life these days. Today, we'll meet the newest member of MDI Bio Lab's team of investigators, Halyna Shcherbata. Hi, Halyna, how are you?
Thank you. I am good, excited.
Okay, good. So are we. Just a couple of words about Dr. Shcherbata. She's a native of Ukraine. She earned her Ph.D. at the Lviv National University of Ivan-Frankov in 1996. Since then, she's held positions at the University of Washington, the Max Planck Institute for Biophysical Chemistry in Göttingen, and most recently, Hanover Medical School. She joined the MDI Bio Lab team, I think it was in 2022. Is that correct? Or did you get here a little bit earlier, Halyna? Archer.
Yes, I came the first time in 22.
Yeah, in 22. She's published numerous research articles that bear down on the molecular mechanics of health and disease. She's a leader in the use of Drosophila melanogaster, the common fruit fly, for her biomolecular studies, and we'll get into that a little bit later. She's brought those flies back into the laboratory stable of animal models for comparative biology. They were here a while ago, I think, with Vicky Lozic, Dr. Vicky Lozic, but then they weren't part of our stable, and now they're back thanks to Dr. Shcherbata. And to ask a little bit about how that came about, we turn to the president of the company, Dr. Herman Haller. Dr. Haller, how are you today?
Thanks, I'm well.
Nice to see you back in town after a quick trip, I heard, abroad. So tell us a little bit about why you wanted Dr. Shcherbata here at MDI Bio Lab. How did you get to know her, or how did you get to know her work, and what did you think made it a good match?
Yeah, I'll be very brief. Thank you very much for the question. I'll be very brief because I'm much more interested how Halyna sees this and how she actually came about to join MDI Bio. But very briefly, I mean, I've known Halyna for quite a while. She's a famous researcher both on Drosophila and RNA. In the meantime, I've learned that there is more than RNA. By the way, the fruit fly, which you see here, this fancy picture, this is not Marcopendid, but this is Drosophila. This is the fruit fly. And so Halyna is part of the faculty of the graduate school at Hanover Medical School. And everything in science is about communication. Everything is about you meet people, you talk to people. And I could first see this link between Halyna's work in MDI-BL when we had this meeting in Hanover, where our faculty met the graduate school of Hanover Medical School. And there was all of a sudden Dustin Updike talking next to Halyna. And it was obvious that, to be honest, we never understand really what Dustin had been doing. I mean, he's doing RNA work and these liquid organelles and stem cell lines and all that. But you could see the sparks flying between Halyna's work and what Dustin is doing, and that it became obvious it would be great to have Halyna at MDI BL to strengthen this research. And then she came to MDI BL, and from there on, she has to tell us what she thought, what she thinks about MDI BL, and how she thinks this will work out.
Thank you so much for great introduction to both of you. Oh, this is a very interesting question, what I think of MDI-BL, because when the first time I came here, I completely fell in love with everything. Not only the surroundings, the nature, of course, is beautiful, but also people, the scientific atmosphere. That was all really, really different and really great. And then I felt right away I would like to be a part of this community, and I would like to contribute as much as I can to make this place even better, and to just be a part of such a great group of people, scientists, researchers, all the team that is here. Everyone here is really, really excellent. And I thought that our research would fit to what is done here, because we do a lot of different things, because we are trying to model human diseases in a Drosophila model. And we have worked on several human diseases, and we are trying to find at the molecular level, at the level of cells and tissue and signals, what is going wrong, and how can we deeper understand the cause of the disease and maybe help in finding some new therapies for the disease. And now I will just introduce you to my favorite organism, of course, which is a Drosophila. It's a beautiful genetic model organism. It looks great because it's yellow, not blue like here. It has beautiful red eyes, or brown, or vermilion, or whatever, cinnabar, different eye colors. And Drosophila, if I can advance my slides.
Dr. Shcherbata, while you do that, I'll just mention to people who tuned in that you should feel welcome to ask questions. You could use the chat function on Zoom. You could also raise your hand at the bottom, and Jerry Bowers or I will try to get to your question. Finally, if you have a really pressing question, and that's what we're all about here, is pressing questions, just go ahead and unmute yourself and ask away. So sorry to interrupt you, Dr. Shcherbata. Please tell us all about why the fly.
The Drosophila is used since 1950s for the genetic research. And I think it was just an accident, or a very lucky accident for all of us, because the Drosophila that we are using in our research has only 4 pairs of chromosomes. And for example, humans have 23 pairs of chromosomes, which of course makes genetics a lot more complicated. But not all Drosophilas have only 4 pairs of chromosomes. The one that was chosen by accident actually is the best genetic model system. So in Drosophila, there are different types of development. So there is embryonic development like in humans, and then there is this stage that is called larval stage. And we think that this is like a childhood, and then there are some hormonal changes that are happening, and we arrive to pupal stages. And this is the stage where the stage of metamorphosis, and those are teenage age. So embryonic is period, it lasts only 1 day. Larval stages, 5 days. Pupal stages, additional 5 days, which means that after 10 days, we have a new adult fly that can then generate a lot of progeny. One female can lay up to 300 eggs per day. And so we keep flies in those beautiful vials with the food that is suitable for humans. And then all this makes flies a very, very cheap model that gives rise to a lot of progeny in a very short period of time. And what is very important now that we do not need any special work permits to work on these animals, because for vertebrate animals, it requires a lot of additional documentation. Having flies for so many years as a model organism, researchers generated multiple genetic tools. We can do with flies anything we want. We have mutations in every gene almost. We can overexpress in a tissue-specific manner different genes and proteins. And I would say that right now, the genome of Drosophila is one of the best studied genomes that we have, best annotated genomes that we have. Also, flies, even though they don't look like humans, but they have the same body organization as humans. They have all major organs and tissue that humans have. And there is a lot of complexity because it's a living organism, pretty complex, that has also complex behaviors. And the most important for us as geneticists that are modeling human disorders is that 70% of all human disease-related genes are conserved in Drosophila. And all this together, of course, makes this organism as a really, really great system to study human disorders. So here I'm showing you, for example, this is the fly, and here we have indirect flight muscles. They are called indirect because the movement is not done by wings directly, but when those muscles contract, then the chitin that is around the fly is changing this shape, and then wings can beat, and then the fly can fly. And you know that flies are flying really, really quick, and those muscles are one of the fastest muscle types in the entire animal world. So they beat 200 times per second. And these muscles have a structure that is very similar to human fast muscles. And we can dissect those muscles. We can make slides of these muscles. And then, as you can see here, we can analyze how the muscles look like. We can measure their size and shape and see if we have a mutant that muscles, for example, are atrophic or start to degenerate with age. So, and this provides a really great system to study muscle diseases. We also can do different genetic experiments to study behavior. For example, here we have two types of flies. The wild-type flies, and those flies like to climb up because they have positive phototaxis and negative geotaxis. But for example, here we have dystrophic flies, and these flies cannot really climb all the way to the top of the vial. And by doing such a simple experiment, we can measure climbing index and see and kind of assay fly mobility. And there are also additional assays. So for example, we can do learning assays for flies. We can do memory, sleep assays, whatever you wish. Everything is easy and it's quantifiable and it's repeatable. One of the projects that we have in our lab is to study neuromuscular dystrophies in humans. And so here we work with such complex, which is called dystrophin glycoprotein complex. And the key component of this complex here is a transmembrane protein dystroglycan that connects the extracellular matrix to the actin cytoskeleton via dystrophin. And the mutations in this complex in humans are associated with muscular dystrophies. Mutations in dystrophin in particular lead to Duchenne and Becker muscular dystrophies. And these dystrophies are only kind of developing after, and this is X-linked disease, so it's mostly boys who have those diseases, and they are developing after 9 years old. And interestingly, the kids that are born with deficiencies in this dystrophin gene, they are even more active than their siblings or the kids of the same age. But when their muscles are participating in physical activities, then of course they are damaged, like normally muscles are damaged. And normally muscles regenerate, and their muscles also regenerate to a certain age very actively. Therefore, they are doing okay. But then at one point, muscle stem cells stop dividing, and there is no muscle regeneration anymore. And so this leads to mobility defects, and then for Duchenne muscular dystrophies, by the age of 30, all patients die because their heart cannot beat and their diaphragm is not working. Becker is a little bit milder muscular dystrophy, and there are no cure for now, even though gene therapies and microRNA therapies are proposed and tested now. Mutations in dystroglycan that binds dystrophin cause another group of muscular dystrophies that are called dystroglycanopathies, and those, in addition to muscle defects, also have defects in the brain and eye. And there is like a huge group of those disorders that differ by their appearance and severity. And there is Fukuyama, for example, muscle eye brain disease, congenital muscular dystrophy, limb girdle muscular dystrophy, and so on. And these dystrophies are actually, they are very rare, but if you put them all together, it's also a large group of neuromuscular disorders. And unfortunately, as of today, we can only properly diagnose 30% of them, because the rest are associated with mutations in some proteins that are associated with this complex, but we do not know about them yet. And therefore, for proper diagnostics, we must understand a little bit better what this complex is doing and what components of this complex are acting in the muscle or in the brain. So we've been working on this complex for many years, since 2007, when I was in Seattle, and we established the muscular dystrophy model. And ever since, we are kind of trying to figure out what the complex is doing, what new we can learn, and do we have a new target for therapeutics. So
Dr. Shcherbata, I counted, I think, 16 or 17 research articles there, and those are all articles that you were an author of, and often the lead author, I imagine, which, using the fly model, described components of these various dystrophies that are common to both flies and potentially to humans. Is that correct?
Not potentially, definitely.
Definitely. Okay. All right. So
in some of those
real
papers,
progress.
we also used human cells and human neurons and human muscles and mouse muscles. So it's always a combination. This is what we try always to add human component to our research to make it more relevant, let's say. What is important also to say here is that it's not only relevant to muscular dystrophies, the levels of these factors decrease with age. And you know that there is age-dependent muscle wasting and also cachexia after cancers, where muscles are atrophy is happening. And then this also is important to understand why with age the dystroglycan levels go down and how we can prevent this to keep our muscle healthier in aging.
And you mentioned that some of these diseases are quite rare. How many flies does it take to run a study on any given, molecular or genetic level program that
A
you're trying to
lot.
look at? How many do you have at the lab now?
It's we do not count
You don't
flies
count them.
because they are so kind of easy to get.
Mhm.
Millions. It's like during all those years, then millions. But in the lab, we keep in stocks only flies with a certain genotype that we do know, certain mutants and so on. And so we have 2 homes, 2 vials per each genotype in the stocks.
Okay.
But then when we do experiments, then we extend this, and of course, then we treat them and age them and train them and yes, and dissect their organs. But a lot more than we can do with any other model organism, except for C. elegans, probably.
And so some of them, when you manipulate them genetically, they're manipulated to be susceptible to these dystrophies at times. Is that?
They are mutants already.
Yes.
So they already have a mutation in the gene that is responsible for dystrophin or dystroglycan or any other component.
Okay, thank you very much.
Then what we can do in flies, what we cannot do in humans, and this is maybe a complicated scheme, but it's just saying that we can find what are the components of this complex in the muscle and what components are present in the brain of flies. And then kind of divide this because we have phenotypes different in the muscles and in the brain. And this is also very important because if we want to modulate or change something in human organism, we really need to know what is going on and what is the function in the cell where we want to target dystrophin or dystroglycan. And by doing these different experiments, we found in our mutant flies this beautiful phenotype. So when we looked at the brain of dystroglycan mutants, we found those lumps that are kind of outgrowing the brain shape. Normally, the brain is smooth. And when we saw this for the first time, we said, oh my God, those must be cancers. But then we do all the tests, and those are not cancers. Those are just neurons that are pushed outside of the brain because they express different levels of cell adhesion molecules that are controlled by dystroglycan. And we were wondering, what is this phenotype? And then we went to the literature and were reading and looking, and we found that actually mutations in dystroglycan in humans lead to this very peculiar phenotype that is called cobblestone brain, because the brain surface looks like cobblestones. And this is actually lysencephaly type 2 that is associated with abnormalities in dystroglycan glycosylation. So this is really a good model fly to have to study dystroglycan function in the brain. And here you can see that in the normal brain at the cortex, neurons that are generated by neuronal stem cells, they migrate and then they make different layers. And in dystroglycan mutants, these layers are not properly organized. And those neurons, similarly like we see in Drosophila, are breaching glial limitants and make those cobblestones. And of course, this is an awful disease, and most of cobblestone type 2 lysencephaly patients die during the first year or during embryonic even stages. So then, having this model, we studied a lot about function of dystroglycan in Drosophila, and also looked how can it be applied to humans. But we realized that humans are actually very bad model to study what's going on in the brain. First of all, that humans have 86 billion neurons in the brain. And in flies, we have only 135,000. So it's a lot better. Then humans have brain compartmentalized, and there are different parts of the brain that are responsible for different behaviors, for example, movement or learning or smell. The same we have in flies. The brain is compartmentalized and we have the same structures. And of course, in flies, we can study different behaviors. And most importantly, we can age flies quickly, and we can treat them in different ways and have good controls, because humans don't want to behave properly or eat all the time the same food, or like have day and night conditions controlled like we have for the flies. So really, if you think about those comparisons, that at the molecular level, at the level of the cell, fly is a lot better model. And
how do you age a fly?
just keep them for longer time in those beautiful homes, so they can live there for up to 70 days. So, but normally after 30 days, we consider that the flies are older a little bit. And it's easy. It really allows to do all this aging research a lot faster. You heard the last time Anastasia talking about the killifish that is aged in 70 days like flies, which is a great model, but complicated and expensive, and we really kind of can have a lot like better quantity, I mean, the experience to work with aging animals. And aging now is a very kind of hot topic because of the aging societies, and the whole point of having older societies is to have a healthier age population, not to have people that live longer, but people that live longer and are healthy. And the problem of neurodegeneration is a really big problem in aging societies because, first of all, right now there are more than 55 million people that have dementia worldwide, and we have 10 million new cases every year. And dementia is the seventh leading cause of death and the major cause of disability and dependency among older people globally. And in 2019, all data, but dementia costed the economies 1.3 trillion US dollars. And there is no cure for dementia. And what we are now, when doing all this aging research, we are trying to understand why the brain is aging. Can we prevent this somehow? And can we use some kind of treatment to prevent aging of the brain? And normally, when you think about causes that lead to neurodegenerative diseases, some of them are genetic and some of them are toxicity-induced degenerations. And there is this one gene that is called human neuropathy target esterase, or NTE, and mutations in this gene cause a lot of different conditions, spastic paraplegia, ataxia, and a lot of different syndromes depending on where the mutation is. But also, when this gene is affected by chemicals, then toxicity-induced neurodegeneration occurs that has the same phenotypes or the same conditions as genetically induced diseases. And how do we know that this is also toxicity induced? This comes all the way to the time of prohibition, and this is Jamaica ginger that was sold in drugstores because it contained 80% of alcohol, and it was used as a kind of officially allowed way to get alcohol. Because it contains such a high concentration of alcohol, for its to keep it so concentrated, certain chemicals were used. And one of those chemicals was actually the neurotoxin, but nobody knew that this is a neurotoxin. It's organophosphate component that allowed to concentrate alcohol. And many people who had this Jamaica ginger, that also was called like slang Jake, developed paralysis of the upper and lower limbs. And this paralysis was like a very kind of obvious how people move. And then, of course, researchers started to look why people have this disease. And it appeared that this organophosphate actually blocks neuropathy target esterase, and it causes the same phenotypes and the same problems with paralysis of motor neurons as a mutation in neuropathy target esterase. I just want to say that the same compound is used, was used until the latest times for as herbicide and insecticide in agriculture. And yes, and I hope that now we know more about toxins, and probably it's a little bit more controlled, that we would not develop neuropathies because of the chemicals that are used for like normal life without consideration about the causes that they can do. So why we studied this neuropathy target esterase? Because in flies there is ortholog of this gene, and when it's mutated, it causes brain degeneration, and as you can see, those holes in older flies. And it's called Swiss cheese in flies because we like nice names, and the holes that we see in the brain are similar to the holes in Swiss cheese. And we didn't know when the gene was described in 1997 that it is a human neuropathy target esterase ortholog. And so when we started to look at this gene, and I also work on this gene from 1997, we found that this Swiss cheese protein is expressed a lot at the surface of the brain. And the surface of the brain is actually blood-brain barrier. And all organisms with complex nervous system develop this barrier to isolate their neurons from blood and protect their neurons, their brains, from toxins and other substances that can be in the blood. And for example, in a healthy brain, this barrier is unpermeable, and only certain components or very small substances can enter the brain. And you know that it also protects us for multiple drugs that we are using, and only some drugs can penetrate the brain. And it was shown that, for example, in Alzheimer's brain, the blood-brain barrier is broken, and then, of course, this induces inflammation in the brain and kind of promotes even to develop Alzheimer's disease even more. And so what we do in flies to study if the blood-brain barrier is permeable or not, so we inject flies peritoneally with the, here was a dye that we can see later and let them recover for more than 12 hours. And after 12 hours, we dissect their brains, and then we look if dextran dye penetrated the brain. And as you can see here in controls, the dye stays outside. This blood-brain barrier is not permeable for the dye. But for example, in Swiss cheese mutants, the dye goes inside the brain easily, which means that this blood-brain barrier is broken. And then we looked more carefully what is happening. And then when you see here in control, and in green is marked the barrier per se, that this is a very smooth and nicely organized line. And in Swiss cheese mutants, there are all those kind of wraps and aggregates, and you see that the brain blood barrier is broken. So we were trying to rescue this phenotype by overexpressing Swiss cheese in those cells, or overexpressing human neuropathy target esterase in those cells. And we found that expression of human neuropathy target esterase fully rescues this phenotype, suggesting that the function of Drosophila Swiss cheese and human neuropathy target esterase in the blood-brain barrier is evolutionarily conserved.
Dr. Shcherbata, when you say rescue the phenotype, what does that mean?
It means that it goes all the way to normal.
Okay, got
So
it. It's returned to normal.
Right. So instead of being like this, it looks like in control. So it goes all the way to normal. And then we wanted kind of to understand, why do we have holes in this brain? Why do neurons inside the brain die when we have abnormal barrier? And we suggested that maybe inflammation is causing the death of the neurons. And to test for this hypothesis, we fed our flies with normal drugs, the drugs that are used very often, sodium salicylate, it's like paracetamol and rapamycin. And we found that those anti-inflammatory drugs can rescue this phenotype in the brain and kind of can protect blood-brain barrier, which is really exciting because this not only explains what is the cause of the disease, but also shows that it can be treated by commonly used drugs. And just to finish this part of the talk, right now the term inflammaging is a very kind of important topic for the discussion, because during aging, the organism is not kind of suppressing inflammatory responses active, like efficiently. And chronic inflammation, which is like low rate inflammation, sterile inflammation, is going on for a very long time. And this is called inflammaging. And now what we find that this inflammation is leading to blood barrier dysfunction, but also blood barrier dysfunction causes inflammation. And this is this vicious circle that then makes brain cells to degenerate.
Dr. Shcherbata, I noticed that in the illustrations you had, or the microscopy that you had of the various versions of the fly brain and how its blood barrier was structured, it looked like it was dated in 2023 when you were looking at how different substances could repair problems with that blood barrier. Was that when the research was done, or is that just when that photo was from? So I'm curious if they're starting to be used.
So we submitted the paper in 2023 and put it on bioRxiv, and it has been reviewed, and I hope to resubmit it today. Will not happen, hopefully by the end of the week. So the reviewers' responses were very positive, and we did everything that we could, and they asked for, and which they asked for really good experiments, and hopefully that 2024 will be the year when it's published.
So this is really cutting edge.
Yes.
And I'm curious, so it's maybe too early to see some of these insights playing out in a medical or clinical or therapeutic environment, but there's clearly an implication there. Is that fair to
Yes,
say?
so this paper is even more interesting because we showed so this all degeneration of the neurons was it's not clear why it happens, but it was only associated with like spastic paraplegia, for example, and syndromes that are kind of neuropathy target esterase related. But what we found that in those cells that are making the blood-brain barrier, actually lysosomes, which are responsible for trafficking of everything within the cell, they are abnormal. And our study leads neuropathy target esterase to lysosomal storage disorders. And lysosomal storage disorders is a huge group of more than 70 metabolic disorders that are associated with abnormal lysosomal functioning. And they are pretty often, depending on the population and type of the disorder, they can be with the frequency of 1 in 800. And so this work leads even kind of connects neuropathy target esterase associated diseases with a larger group of metabolic diseases.
I see that Dr. Haller has a question or a comment. Dr. Haller, please jump in.
Yes, I've a comment. Thank you very much. I mean, this is fascinating, Halyna. I'm enjoying this very much. I knew part of this, but all the time it's new things. And I'd like to remind us all, on the one hand, we have working on inflammation. Sam Beck published a very important paper that the genetic footprint of aging and inflammation is very comparable. So it's exactly down the line what Halyna just described. It's not that far off. I mean, we are treating and we need to treat inflammation in aging as well, especially chronic inflammation. But then Halyna talked about blood-brain barrier. This is endothelial cells. I immediately react to that because we are working on permeability and changes of permeability in endothelial cells. So that fits very nicely. And first of all, it's a wonderful arena for discussions. We'll have retreat in 3 days, and I'm looking forward to exchange then views on how we could collaborate on that. And there are others at the Institute, and they are interested in this as well. So it's wonderful to start these discussions using Drosophila because it's so fast. Halyna described this. Compared to our other models, I mean, this is like science fiction. You do a mutation and you see something within a couple of days. Sorry.
Even better, we have a mutation. We have mutations already in most of the genes.
So we have another question, and Dr. Haller mentioned that there's a retreat coming up, and it's a really nice example of the cross-pollination that's really special about this organization and the various models that are used that lead to allied paths of inquiry. So it's always a creative moment. So somebody at the watch party at Anna Maynard's house in Southwest Harbor for this Science Cafe has asked a question or has raised their hand anyhow. And of course, I can't tell who it might be, but let's please, whoever it is, pipe up.
Yeah, I just had a question whether you ever use DMSO, dimethyl sulfoxide, in your experiments, it being a very powerful solvent and passes the blood-brain barrier.
Yes, so we use this as a solvent for, as you know, for many chemicals. And but we never tested if it affects blood-brain barrier. We used it for mutagenesis, but it's a good point. It is, I think it is permeable, right? It's the molecule that can go through, because in humans, the molecules from 50 to 500 kilodalton can potentially enter the brain, and especially lipids allow a better entrance into the brain. Also, human brain barrier is not completely not permeable, right? We know that narcotics or other drugs enter easily the brain. And also during night, it's more permeable than during the day, because the brain has to clean itself and also kind of release products of metabolism to the blood. And therefore, it also is a very important point now. It was discovered in Drosophila that there is a fluctuation in blood-brain permeability during day and night. And it's pretty new, and therefore it adds additional kind of information about when and how you should use your drugs at night, in the morning. Would it affect your brain or not? Because many drugs would be toxic for neurons, I think.
Yeah.
And now,
Widely in concussion cases in athletics because of that capability as a solvent and passing the blood-brain barrier.
Yes, I have to look into this. It's a good point, yeah.
so we're coming up towards the end of the time, and so please, if you have more questions that have been tickling at the back of your brain, bring them forward, and either through the chat or raising your hand. And well, Jerry Bowers has just asked, ready to ask one.
Halyna, thank you so much. That was a really fascinating presentation. You know, you mentioned, you sort of did the comparison between the complexity of the human brain and, you know, maybe relative lack of complexity in the fruit fly. And I'm just curious, do you anticipate that it will be more challenging to sort of address this in either a mouse model or a human model? How are you anticipating that will go when you try to replicate this kind of thing in another model?
So if we talk at the level of what are the components of the brain, then of course there are neurons, right, that communicate and send signals and so on, and kind of control our behavior. And there are different types of glia cells that are providing neurons with metabolites, protecting them, and making all those different parts of our brain. And in Drosophila, there are neurons and there are glia cells, and many types of glial cells, similarly to human brain. And then the synapses are also similar. Receptors, neurotransmitters are somewhat different, but also the same. Many of them are the same. So it still is a very good model. We do not have a complexity of the behavior, even though flies are behaving very complex. So try to catch a fly. It's not so easy. They are fast, they are good. And some flies, insects are flying multiple like miles to find something, and so their behavior is also complex, but it's just it's not comparable and comparable. At the level of the tissue structure and cells, and so it's comparable. But then, of course, at the level of the behavior, it's different. And mouse models are very good, but they are also, they require a lot longer time to study something. And aging especially, and inflammation, and it's a lot more expensive too.
Yeah, no, I appreciate that. Thank you.
Dr. Shcherbata, I'd like to ask you, where are you headed next? I know you've launched new research projects on Mount Desert Island. You're waiting for data to come in and to really be trued out before you say a lot about it. But what can you tell us about what you're interested in now? What's got you fascinated that you're working on at the lab in the coming months?
So there is another very interesting project that is related to human neurodegeneration.
Dr. Shcherbata may be muted. I think you might have hit your mute.
You had me on mute. Where was I?
You had just started when
Okay.
we lost you.
So right. So there is another project that we are working on and would like to work here that is associated with multiple neurodegenerative neuronal disorders such as attention deficit disorder, the ataxia, spinocerebellar ataxia, and also with diabetes and hand osteoarthritis and heart defects. And this protein is an RNA binding protein that also is associated with those different granules that Herman mentioned before that Dustin is working on. And so we will model diseases associated with RBFOX1 in Drosophila, and we will try to figure out how one protein can be associated with so many seemingly unrelated disorders and have different phenotypes. And yes, so this would be our project here.
You once said to me that the number of different things this particular protein does is mind-blowing. So I think you're going to be having a good time. Dr. Haller, please, I think you wanted to weigh in with the point as well. If you can unmute yourself.
Yes, I think I would like to make one remark, because Cherry was asking the question about what about other models? And I think it's really very important to understand what MDI-BL has been doing for 125 years. We are using a model, exactly what Halyna described. We were identifying the molecular mechanism in our model, which we think, and we have lots of evidence for that, is important for humans. And then you go from our model directly to the human. This is what we are trying to convince now biomedical and pharmaceutical industry, because it may seem that the mouse is more human than the fruit fly, but that's not the case. So these model system, and some of you may remember, we have found the mechanisms how the kidney in humans work in the shark. And we were famous for that. We are still famous for that. And now we are becoming famous for neurodegenerative disease in Drosophila and in our other animal models. So it's not that we are doing this on a very basic level and then we go gradually upwards and finally we end up in the mouse or in dogs, etc. No, we are identifying a molecular mechanism and then we go directly into humans. And that's the beauty of what we have been doing for a long time, and we're still famous for that.
Yeah, I agree. It's just you don't need to have all the models in between. You have now with organoids, with human cell culture, with everything, we can go directly to human, and it's a lot faster, just time-wise, and which is very important for graduate students. That if you have your aging animal, then you want this animal to be aged several times during your PhD, not only once. I'm just saying that mouse models for aging are difficult because they live for 5-7 years. And I'm not even talking about other model organisms that live up to 70 years, like yes. I can't. Fred,
Are there other questions or comments that people would like to bring to the table at this point? Some will enjoy the opportunity to talk with Dr. Shcherbata more tonight. But in the meantime, I'm not seeing any more questions right now. Dr. Shcherbata, anything else you want to add just before I start pitching the next Science Cafe?
I'm just really, really excited to be here. Like everything here is just unbelievable. And thank you so much for inviting me and for having me. And just thank you.
It's really fascinating, wonderful to peer into this world that you're working in. It's just incredible. And thank you so much for your time. And I want to thank everybody who participated. Really great to see you guys check in with us each month as we do these science cafes and find out a little bit more about the amazing things that our researchers are up to. And you'll probably want to next month. Our guest will be Dr. Ed Benz, Jr., who is the CEO Emeritus of Dana-Farber Cancer Institute. He's also Vice Chair of our Board of Trustees. And he happens to be an expert in sickle cell diseases and other blood disorders. And he is the director of the NIH's Sickle Cell Initiative. And this comes at a very interesting moment when gene therapy is starting to reach towards its translational potential and its real world application. Late last year, the FDA approved a gene therapy for sickle cell disease. And so Dr. Benz will be with us to talk about that and about the promise and maybe the challenges of using CRISPR and other methods to manipulate genes to create human therapies. So I think you'll all see, if you've signed up for our e-news or for our science cafes, you'll get an invite to that. There'll also be soon online recording of this event that will get to you, the links for that via email. And so keep an eye on your inbox for those. Jerry, is there anything you'd like to mention just before we sign off and you take control for that?
No, just to express my appreciation to Dr. Shcherbata and all of you for joining us this evening, and we look forward to seeing you next month.
Bye.
Thank you so